Okay. Thank you very much for having me here today. Thanks so much for the organizers and putting all this on, especially all the translators and the students working so hard to make sure everyone can speak to one another. It is Thanksgiving in the United States, and despite its sordid history, it is a time to reflect on what we're thankful for. So I'm very thankful to be here with all of you on today. Okay, great. So I am an associate professor in anthropology at Penn State. I also head the ancient DNA laboratory there. I am a microbiologist by training, so I'm not an anthropologist, I'm not an archaeologist. I really like microbes, and I like what they can bring to the table when we think about archaeological and anthropological questions. So we have been working on a project over the last several years to bring microbes to the conversation in the Pacific. And I think quite often we think about migrations and the pattern of this migration and the origin of these migrations. And that is where our thoughts started, although I will elaborate later on where our thoughts are now and sort of where they're headed in the future. And so there's been a ton of amazing research that's really gone into answering these questions over time. We've heard so much about it already, I think, at this conference. And I think we're all trying to use both different proxies as well as direct evidence to really understand these migratory patterns. And we're realizing that they're not straightforward, they're not simple, they're not easy, right? And so despite, I think, some of our thoughts early on about what this might look like, we're hoping that we can add new lines of evidence to better understand these patterns and timing. And that's really where microbes can come into the picture. And I'm not the first person to propose we use microbes to track migrations or to track mobility of people. In fact, this has been done for a while now. And one of the first studies was in 2003, where they used Helicobacter pylori. This is a microbe that lives in your stomach. It's very acid-loving, and it's hereditary. So it's handed down to you from your parents through the generations. And so it can be used to track human migrations over time. In fact, it does have very nice geographic patterns across the globe, which may allow us to associate that with where humans have been and where they've come from. The early study in 2003 didn't use full genomes, but again, it's been elaborated on over time. And in 2009, they were able to use more genomic data from this exact same species to really start to get at some of these Polynesian questions of migration and expansion. And it's interesting here, they find that they have sort of one wave of microbes that are associated with the peopling of Sahul, and they have another wave of microbes associated with the Austronesian expansion. But this was all done in modern individuals, and as we know, things in modern day can be quite messy, especially considering what's happened with Europeans in the area. And so using ancient DNA may be a way to go back in time and actually look at the microbes that people had at a given point and use those microbes as a way to mark or to trace migrations through time. And we're doing this using calcified dental plaque or dental calculus. This is a complex bacterial biofilm that forms and grows on your teeth. In fact, if you had some of the Tahitian cola at lunch, you may have felt that slime on your teeth growing over time. Those are microbes reproducing and living very happily on your teeth. And if you don't brush your teeth, or if you don't have access to modern dentistry, those will form into a rock-hard like matrix called dental calculus. In fact, the dentist using its little sonication machine, the bzzz in your mouth, they're actually breaking that dental calculus off of your teeth, okay? So it's a very normal biological process these days that we have dental calculus, and it really calcifies and preserves microbes within our bodies in real time while someone is still alive, which is also very important. If we're thinking about microbes being everywhere in soils and water, it's important these microorganisms are trapped from the human in time, and that we can really be sure that they are associated with the human and not necessarily with environmental contexts. So sequencing DNA preserved within dental calculus has become our bread and butter, and it's become a way for us to be able to trace which microbes people had in the past, at least in their mouths, and develop new tools to understand how we might use these microbes in archaeological and anthropological contexts. And in fact, in 2016, we proposed this method of using oral microbes rather than our stomach microbes to track human migrations. And we wanted to use microbes that are passed, again, from mother to child and passed through the generations that might act much like human DNA acts. But the nice thing about calculus is you don't just get one microbe, you actually get a whole suite of microbes. And we refer to those as our microbiota. And this could include bacteria, fungi, viruses, also archaea, protists. If it's small and under a microscope, it will probably be there and we'll find it. To give you some perspective here, there are about 200 species in every single person's mouth. Okay, so you have 200 species of microbes in your mouths right now. So that's a lot of diversity to try to track through time. Lots of opportunity here. But within every single person's body, we have about 1,800 species. And in fact, you carry about 100 trillion bacterial cells with you. That's equivalent to about 3 pounds of body weight or 1.4 kilos, which is analogous to the weight of your brain. So you have a whole extra organs worth of microbes floating around in your body. But it's their genetic information and the functions that they carry with them that are really important for us. Because in fact, the genetic information encased in these microbes outnumbers the human genome 100 to 1. 100 to 1. So 99% of the genetic information inside of your body is not human, it's actually microbial. So if we want to understand how humans have come to be, where they've been, how they're adapting to unique environments, we have to understand the other 99% of the DNA within the human body. And the wonderful part about these microbes is they change, they are altered in some states. Some of them are changed and altered, some of them are directly inherited. And so we can use them to also understand a lot about our diet, a lot about our environmental context, and a lot about health and disease. So very, very useful data indeed. So in our project here in Central East Polynesia, we really wanted to ask 2 key questions using dental calculus. The first one was, can we detect human migration signatures using oral microbes? And this has not been done before. And so we really wanted to ask, can we even do this? Is this really something that checks out? And secondly, how do oral microbiota adapt to specific environments? As people settled in new locations, did they adapt biologically to these new places and new diets that they may have encountered over time? So to do this, we really kind of re-envisioned this project more recently into a very community-grounded approach. We want to work with local archaeologists and local community members to do this research. And so today I'll talk largely about data that comes out of Central East Polynesia from work here with many collaborators sitting in this room. And I've also added in data from Palau and New Zealand or Aotearoa as a sort of comparative data set so you can understand and have some sort of view of what's going on. So let's start with the first question. Can we detect human migration signatures using specifically ancient oral microbes, but even modern ones? And the short answer is, first, you have to actually identify which microbes are present across all people in the Pacific, and are they abundant enough, are they there in enough abundance to be able to get enough DNA to reconstruct full microbial genomes? The short answer is there are 12 particular bacterial species that we find present across all of these individuals, and that we find in high enough abundance to reconstruct genomes with coverage that we would trust to then push forward into a phylogenomic approach where we're looking at the sort of evolutionary history or tracing the history of people. And in fact, this species here at the bottom in blue, Anoraliniaceae, bacterium oral taxa 439. I didn't make this name up, but it's dealing with bacteria, it's awful sometimes. But anyway, this species is the most abundant, and so we've focused a lot of our work on this particular taxa. And so this is one of the first trees that we generated, and this also leveraged data that was published in the literature from Africa and Europe and Japan, again, as reference populations. And we took a really conservative approach when we made this particular tree to really make sure that we were indeed seeing the right signatures. And in fact, we do see very large-scale human migratory patterns coming through in the presence of this microorganism. That's important because previous research had only seen it present in sort of demographic transitions. They'd been able to track differences here. But here we can actually track differences as people would have left Africa and as they moved from the Middle East and Europe, elsewhere in the world. The first interesting finding from this analysis is that individuals from Palau, and these are all ancient individuals, that individuals from Palau actually separate from Japan. And that are not sort of necessarily ancestral to all the microbes that we find in Polynesia. That was really fascinating to us because it may suggest that there's some sort of interaction with another thank you, 5 minutes, with some other earlier population. Now, I'm not suggesting that Polynesians are derived from Japan, just suggests that there were mixing of microbes sometime earlier in the history. So that's a first very interesting observation. The second one is that in this particular study, we were working with Maori individuals, and one of their questions was in New Zealand was really to look at whether or not these sort of large scale patterns across different continents might be used for repatriation. Indeed, if we look at this tree, we actually find all of our Maori ancestors there on the bottom of the graph, clustered at the bottom of Polynesia. Whereas one individual that was believed to potentially be of European nature, but was sitting in a New Zealand museum, we actually find that their oral microbes appear to cluster with Europeans, suggesting that these very large scale patterns that we find in oral microbial genomes may actually be used for less invasive repatriation methods using this approach. And last but not least, to talk about what's going on in Central East Polynesia, we really see a mixture of these signatures. We don't sort of see something that's directly linear, which is probably no surprise considering much of the research that we heard about on day one. We do see that samples from across different archipelagos in this area are largely mixed. So there is no specific sort of linear signal that we're seeing. And this would be indicative of cultures or people in continued contact and continued sort of either trade networks or other interactions. So lots to continue to explore here, and I'm happy to discuss this further, but it's very wonderful to sort of see this mixture of signatures happening here in Polynesia, which we don't see necessarily elsewhere in the world. Okay, so the second question, how do oral microbiota adapt to specific environments? And what might that tell us about biological adaptation? So if we look first at just individuals from Central East Polynesia, we can do sort of clustering based on the overall composition of the microbes in each person. So these are not individual genomes, these are the whole community that we're looking at. Each dot represents an entire microbiome from a single individual. Dots that are closer together are more related, dots that are farther apart are less so. And what we see when we test this using some of our statistical methods is that archipelago and island both significantly explain diversity across Central East Polynesia. And so it suggests that people have different microbiomes in different island sets, as well as different microbiomes in different archipelagos, which is very interesting. We also see that individuals from the Gambier have the sort of most distinct microbiome across Central East Polynesia. If we take a broader approach, though, to kind of put Polynesia in a greater perspective, we actually we can compare these to both individuals from Palau as well as Aotearoa in New Zealand. And we see that New Zealand has very interesting patterning. And in fact, some of the individuals that we find in New Zealand have microbiota that are very similar to people in elsewhere in Polynesia, whereas other individuals have very distinct non-Polynesian-like microbiota. And so there's something very interesting going on in New Zealand, but that's a talk for another day. And we also see that Palau has a very distinct microbiota, say, from what we see in the rest of Polynesia. And again, not necessarily unexpected, but good to see that there is adaptation to Polynesia that's specific to Polynesia, and that it's separate to what we see elsewhere in the island. Perfect. So we really wanted to dig into this further to really see, how many differences do we expect to see across these archipelagos in specific? And so we used random forest analysis to try to say which microbes help us classify each of these different archipelagos. And in fact, just using the microbiota or the microorganisms, we can classify each archipelago with 73% accuracy, which is quite good for microbiome data. And it tells us a little bit about what microbes might be very important in particular areas versus which microbes might not be. And in fact, ancestors from Central East Polynesia are missing some common oral microbes that are highly abundant in Europe at the same time period, okay? And that's interesting for lots of reasons, because some of those microbes can be involved in things like periodontal disease, and it gives us something to look for as we try to compare these individuals to modern individuals. Are European microbes, commensal microbes, introduced during European arrival? We would hypothesize that they are, but it gives us something here to then measure and look at, and then understand how that may influence health. We also see that, just across Central East Polynesia, there are differences as well. So for example, the Society Islands have a microorganism called Fusobacterium nucleatum, which is very much associated with periodontal disease and even abscesses in some points. And that microbe is absent in some of the other archipelagos. So some differential rates of disease that we might see in oral health across the archipelagos may be tied to different microbes that they have for one reason or another. So I've gone through this pretty quickly, a short talk, but I have gone over how oral microbial histories, at least from a single genome of a particular species, in Central East Polynesia are mixed, and this likely signals continued interaction and connection over time, and that these sort of genomes are very different from what we see in Palau. Okay, so there's different microbial signatures that have occurred over time. We also see that whole microbial communities are very specific to archipelagos and particular islands, and these are perhaps a signature of biological adaptation. It'll be very interesting to track how these are associated with health later on. And overall, that microbes provide another lens to really view settlement processes and provide more information about how people adapted and adjusted to daily life. And so our next steps in this project are really to try to understand why some of these differences in microbial communities across archipelagos and across islands exist. And so I've been taking a huge heap of notes while we've been here this week about different data sets that we might be able to collaborate with folks on, about differences in diet, difference in food source, difference in lifestyle changes. So, different habits we might have, or even the difference of different environments on different islands, right? So are you in a sandy place, or are you somewhere up high? And how does that influence our microbiomes? We also want to test if these shifts in oral microbiomes are linked to health or the presence of oral and systemic disease. And so we'll be doing that next. In Europe, we find that specific types of oral microbiomes are more likely to be associated with systemic disease or skeletal signs of disease rather than, say, oral signs of disease. So with that, I will say thank you, and just thank you so much to all the collaborators and local community members that have helped out with the project. It's amazing to work with all of you. Thank you.